When viruses want to attack a living host or cell, they use a very specific protein on the outer surface of its capsid or envelope that will allow its attachment to the surface of the host cell. This specificity is really important, as the virus doesn't want to attach to just anything that comes its way, because it may have no use for it. Hence, having a specific hook that will only attach to a specific target surface receptors will improve its chances of survival. The specific set of cells or entities upon which a virus can successfully attach to is known as its host range, through several mechanisms. Some viruses inject their genomes into the cell by using a needle and syringe type mechanism. Other viruses will actually trick the cell into engulfing the virus into the cell, something called endocytosis. Further still, envelope viruses will undergo a process called membrane fusion, whereby their envelope will fuse with the envelope of the cell. This will allow the virus to enter into the cell without any impediment. Viruses, upon penetration into the cell, will also uncoat themselves by removing their viral capsid, and thereby unmask the nucleic acid inside. This entire process is called uncoating, and again is a process whereupon the viral capsid is removed in order to release the viral nucleic acid into. It turns out that the viral nucleic acid, be it DNA or RNA, will basically take over the host once inside. There are many processes by which this occurs, and this will be covered in later lessons. In general, what you need to grasp now is that the viral genome will take over the host cell in order to copy its own nucleic acid many times over and use the organelles and metabolic processes of the cell to create and assemble the proteins for its capsid. Sometimes the viral genome will actually integrate itself into the host's chromosomes. When the viral genome is integrated into and replicates along with the host's genome, we call this viral genome a provirus. A provirus is a term specific to viruses that infect animal cells. Likewise, when a bacteriophage's genome integrates into and replicates along with the host's genome, bacteriophage is passively replicated along with its host genome. We term it the lysogenic cycle. In this passive cycle, no viruses are produced, and the viral genome is simply copied if the host replicates. This cycle is more colloquially known as a latent infection, and its virus is called a temperate virus. This is when the virus lies dormant